quite rare, but it's cases like this that can lead to major discoveries in trying to locate genes for a disorder. So in this case, what happened was that part of chromosome 20 got attached to chromosome 7. And what happens when this kind of translocation occurs is that at the break point, this gene is disrupted. So we have a gene where there's been a mutation that's occurred, and we have 2 children with autism. So this provides a major clue to perhaps that gene on chromosome 7 that we're looking for, or one of them. And so Enrique and Jerry have now sequenced this gene, and they're looking to see whether mutations in this gene are present in other autism families in our sample. Now, a third approach, and this approach is really quite novel, and the University of Washington, I think, kind of shines in this area, and it has to do with the fact that we have Ellen Weishman here, who is developing the complex mathematics that are required to do what's called quantitative trait locus analysis. Now, the notion here is that susceptibility genes code for proteins that are involved in specific traits rather than full-blown autism. And I think that you've gotten a sense of that from some of the information that I've given you so far. And the notion also is that these traits are not all or none. It's not as if you either have a social impairment or not, but rather, like height or blood pressure, these can be measured on a continuum. So this approach then assumes that susceptibility genes likely have effects on these continuously distributed traits. And the mathematics now are very different in terms of how we do the linkage analysis. This approach has been successfully used recently to establish linkage for reading disability. And let me just show you how it worked. Now, what I've graphed here is the kind of graph that a geneticist enjoys looking at. And what I have here is a chromosome lying on its side, and here are those DNA markers that I mentioned to you. And what we're plotting here is called a LOD score. It's just simply the degree of association between a particular trait and that location on the chromosome. Now, what you see here is that a component process that's very important for learning to read, and that's phonological processing, that's the ability to make sense out of the units of speech, appears to be strongly linked to this region of chromosome 6. Whereas another component of reading ability, the ability to take those units, form words, and recognize words, shows no linkage to chromosome 6. However, if I were to show you a similar graph like this for chromosome 15, what you would see is the reverse case. So what we're seeing here then is the idea that genes are coding for component processes that act like risk factors, and that these come together then to create more and more risk for developing the full-blown syndrome. So right now we are using this approach with our linkage study, and in fact, Ellen has just finished the first analysis of the first 100 families, and I think it's okay to say that our preliminary results are showing positive linkage on chromosome 2. And we're particularly excited about this because there are 2 other groups in the country who have also shown linkage on 2. So we're hopeful that we might be on the right track here. Okay, I'm going to shift topics a little bit here and focus more on what we know about the brain. And I'd like to begin with the autopsy study. So these are studies where we actually look at what's going on at the cellular level in the brain. And these were conducted over 20 years ago, most of these, and they're now being replicated using much more sophisticated technology. But there are 2 findings that stand out. One is that virtually all autopsy studies have shown reduced number of Purkinje cells in the cerebellum. Now, the cerebellum is an area of the brain that participates in almost all the things that we do. It has to do with coordination during language and attention. It's very important for motor control. And the interesting thing about the nature of the abnormalities in Purkinje cells in the cerebellum is that it suggests that things started to go awry prior to 28 to 30 weeks gestation. So it has given us very good evidence that the abnormality in brain development starts really early, even prenatally, before the child is born. Now, the second abnormality that we see in many cases is in the area of what we call the limbic system, particularly the amygdala and the hippocampus. Now, the amygdala is very important for social and emotional processing, and I'll be talking about that some more in a minute. And the hippocampus plays a very important role in learning and memory. So these also give us some hints about why the child with autism has the cognitive and social difficulties that they do. But what do we know, actually, about brain function? What I want to do now is to walk you through a series of brain imaging studies, and I'm going to focus specifically on the social impairment and help you understand why the child with autism has this profound difficulty in relating socially to other people. Now, what we know is that there are several brain regions that are critical for social interaction. And what I have illustrated here is this complex social brain circuitry. And let's just walk through each of these brain regions. The first is this fusiform gyrus. And this is very important for just perceiving faces. In fact, if you have damage to this area of the brain, you lose your ability to recognize another person by their face, whereas your ability to recognize objects and words, process language is fine. A second area is the superior temporal sulcus. This area of the brain is exquisitely sensitive to biological motion, and what I mean by that is detecting eye movements, mouth movements, facial expressions. Then the amygdala pulls all that together and is able to actually recognize emotional expressions. It can detect the difference between a fear face and an angry face and a happy face. And not only does it recognize emotional expression, but it also has to do with your own arousal or emotional response to seeing that other person's emotional expression. So I guess the way to think about this is the fusiform is saying, hey, I recognize that's a face, and oh, it's moving. I see those eyes moving, that mouth moving. Oh my gosh, that face is sad. And then it takes the prefrontal cortex, which develops later, to be able to take this one step further and to say, I wonder why that person is sad. In other words, to be able to speculate about other people's feelings and thoughts and attitudes, to develop what we call a theory of mind. So all of these brain regions have to act in a very coordinated fashion in order for us to interact smoothly and adeptly with other people. In fact, Temple Grandin, who is a very high-functioning woman with autism, a professor, said, you know, I had no idea that other people communicated through subtle eye movements until I happened to read about it in a magazine 5 years ago. And another adult with autism had said, you know, it's as if the whole world knows how to mind read, and I have no idea how to do this. So we use all of this information from the face to interpret the meaning and the attitudes and the feelings of other people. And this ability is critical for social communication. Now, what I'd like to do is to focus on just one of these brain regions. In fact, we're studying all of these brain regions, as are many other people around the world. But I just want to give you a sense of how we try to look to see whether any of these brain regions are actually impaired in autism. And I'm going to only really talk about the simplest one, the fusiform gyrus and face perception. Now, from very early on, humans are naturally interested in other people's faces. It's certainly not something we have to teach babies to do. In fact, young neonates have a preference for tracking any stimulus that has a facial configuration as compared to an equally complex but non-face stimulus. Now, one way of trying to see whether in autism that this face processing system might be disrupted is to use a technique that's called functional magnetic resonance imaging. So this allows us to look at where in the brain is activated when it's processing a specific type of stimulus. It's a measure of the amount of blood flow, which is a good index of how active that part of the brain is. So in a typical fMRI experiment,